I've had this before.
Okay, I'll be back later.
There's a question here, but we can
answer that, it's fine. Yeah, started in direct message.
Yeah, okay. It's okay. We can start.
Okay, back. Welcome back, everyone, after the break. So again, we're gonna, second half of the morning here, I'm gonna talk about some standard operating procedures really related to sampling and standards, and a little bit about how we can measure microorganisms in different samples. And then Torben will give some failure analysis cases after that. Okay. All right. Okay, so when we're assessing any type of sample, I guess, for the possibility of MIC, we really have to have high quality samples, right? So I always think garbage in, bad samples in, garbage out, right? You just won't know exactly, you can't make any sense of any of your data. So no matter what type of analysis you're doing, we're going to focus primarily on microbial or genomics, sort of DNA-based technologies. Getting an answer out about who is there, numbers of cells, things like that, are really only as good as your samples taken. Okay, so big message, you always have to have really nice samples. And so luckily there's actually many, I don't know how to get rid of that here. There's many actually industry standards that have now been developed to assist with proper sampling and analysis for MIC. And I know you can't read any of this, it's very, very small. Torben did share a standard with you in his talk earlier by DNV, recommended practices for sampling for offshore platforms. But there's many different kind of industry organizations that have put out some various standards for at least assessing and sampling properly for microbiologically influenced corrosion. So very exciting right now. This NACE standard TM 0194 was last updated in 2014. Just last week, there was a new revised version that was accepted by the voting members as a new standard, and it's really field monitoring of bacteria. We expanded it, not me, but the scope was expanded from oil and gas to broader industrial systems. So just a lot of different, another standard I'll talk about a little bit later is one related to how to sample and handle samples related to molecular microbiological methods. So it's really, I guess what I'm trying to say is there's a lot of really nice standards that are out there that you can use to follow to make sure you get good quality samples for doing microbiological analyses. So today I want to talk about kind of different methods that we use for microbiological analysis. Couple slides on proper sampling, kind of talking a little bit more about multiple lines of evidence and collecting that type of evidence. And then I want to talk a little bit about how we can do microbial corrosion experiments to see, for example, if we have samples that we're getting from the field that can be quite corrosive. And again, microbes are only one part of MIC. We have to understand those microbes in the context of the metal and the surrounding chemistry. I think in Torben's diagrams, he also has kind of operating conditions, pH, pressure, temperature, etc. That's a little bit kind of folded into the surrounding environmental conditions. Okay, so if we're taking a sample, let's say from a site, a pipe, some a platform anywhere that we think may, the operator thinks there may be some, there's some corrosion, but they want to know if it's related to microorganisms. There's kind of three commonly used and most accepted, I guess, widely used methods for microbiological analysis. So one is called the culture-based, we call them bug bottles, the industry likes to use. I'll kind of go through what those are. That's probably, even though there's a lot of limitations, that's probably the most commonly used method still today. We have a genomics-based, so this is a DNA-based different analyses. So we often call this molecular microbiological methods. So you'll see MMM abbreviated. This is really becoming more common. A lot more people are starting to use these molecular microbiological methods. And that involves extracting DNA. So again, that's an activity you'll be doing today. And then there's also enzyme-based assays, one of which is called the ATP test. So again, you'll be running through that test today, and that's very commonly used, especially in the oil patch, but I also understand it's used quite widely in water and wastewater treatment systems as well. So what I'm going to do is kind of go through these various types of methods so you can kind of see, if you ever have the opportunity to, have a job where you have to investigate for microbial corrosion, what are the things, the tools at your disposal. Okay. So first, we can look at these culture-based bug bottles. And so essentially what you're doing is you're kind of taking some kind of environmental sample and you're putting it into liquid media that has been kind of designed to culture certain types of microorganisms. So sulfate reducers, acid producers, iron-related bacteria, heterotrophic aerobic bacteria. There's all kinds of groups that these types of bug bottles have been designed for. Typically, I think as Torben mentioned earlier, it's usually kind of water-based samples that are used for these techniques. So you would take water from whatever type of system, you put them into these different lab bottles with the media, either to kind of look for indicators of certain types of organisms like sulfate reducers, or you can do serial dilutions to kind of count the number of microbes you might have in your system. You could feasibly take, pipeline residue or pigging debris, which is shown at the bottom here, kind of mix it with water and then kind of use that in serial dilutions. But these are all really liquid-based techniques. And so some advantages of using these bug bottles is they're simple, they're widely used, they give kind of microbial counts. And a lot of times when somebody is monitoring a system for microorganisms, they kind of have a number. We talked about how numbers are really not that important, but that's a KPI for them, a key performance indicator. So let's say you have a water system where you do this kind of bug bottle test, you have 10 to the 6 bacteria per mil, and you have no problems, right? So people like to kind of have that number as like, hey, this is my number of bacteria where I won't have any problems. But now let's say one day you do that test and you have 10 to the 8 or 10 to the 9 bacteria, right? Oh, maybe I'm going to have a microbiological problem, right? So it's kind of used quite widely for baselining and maybe indicating whether you might have a microbial problem. But the main disadvantage is that because this is cultivation-based, you're growing the microbes in really specific media that's designed for really specific types of bacteria. It really only captures a small portion of the microorganisms that are actually in a sample. Okay, so it doesn't really accurately reflect that entire microbial community. So just as an example, a medium designed for sulfate reducers contains lactate and sulfate. So what this does is it only enumerates lactate using sulfate reducers. So it misses out on other sulfate reducers and other sulfide producing microorganisms. So it's kind of got some of that's kind of a big disadvantage, but it is a very commonly used technique. And then what we can do, what we kind of propose doing, because I think we get a broader idea of the microbial community, is to use these molecular microbiological methods or the MMM. So again, we can take the same types of samples, any type of sample really. You can extract the DNA using some protocols, and you can do a couple of different things with that DNA. So you can kind of do something, we talked briefly about this earlier, the 16S ribosomal RNA gene sequencing. It identifies kind of the microbes, tells you the types of microbes you have in your system, and you can actually kind of target some other known genes that give you some sort of function, and you can do some enumerations using these molecular techniques. So that's one thing you can do with that DNA. And the second thing you can do is something called metagenomic sequencing. So this sequences absolutely everything, all the genes in your sample, and you can kind of get a complete genetic potential. So I'm going to just briefly go through what all of these mean. So this is just a basic kind of I guess, description of what you can do with different types of DNA, I guess, or with DNA from a sample. So again, I put a kiwi up there because that's what you guys are going to be working on today. And so you can take anything, any type of sample, and you can kind of run through a procedure to get that DNA out of that sample. And the reason, again, we're using kiwis is that you're going to actually be able to see your DNA today. Whereas if we take, let's say, pigging sludge from a pipeline and try, we know we have DNA, let's say, in our sample, but we can't really see anything. We just have faith that it's there, and we have different tools that we can use to analyze it. But you'll be actually able to see that DNA. So once you have that DNA, again, you can do amplicon sequencing, figure out what the microbes, who the microbes are, or you can kind of do metagenome where you get all of the genetic information about all of the microorganisms in your sample. So just a really fundamental breakdown of these techniques. So for the 16S ribosomal RNA gene sequencing, it's so of the molecular techniques, it's probably the most commonly used method. And it's based on this molecule. It's a 16S ribosomal RNA molecule. It's a highly conserved molecule across all of life, actually. So we have it, every creature on the planet has it. And there's some conserved regions, meaning they have the same number of base pairs, and there's some variable regions. So you can kind of we call it amplifying, kind of a part of this gene for this molecule, and get kind of millions and millions and millions of copies of it. And then we can kind of sequence the ATs, GCs, all the bases that make up that particular region, compare it to databases that have millions and millions of these sequences, and it kind of spits out a list of microorganisms that are in your system, okay? So yeah, you get a list of microbial names. It's not really a quantitative way, but it kind of gives you a relative abundance of certain types of organisms. And so this would be, came from a paper where they were looking at a pipe failure. So you saw that really nice pipe picture that Torben showed with the big thick biofilm on there. And so this was a group that analyzed a pipe cutout that's had a similar sort of biofilm growing on it, and they wanted to know what microbes are in that biofilm that was potentially causing some corrosion of this pipe. And so using 16S ribosomal RNA gene sequencing, they could kind of get a list of the types of microbes that were in the sample. So they found they had iron oxidizers as well as iron reducers. They found sulfur reducers, sulfate reducers, fermenters, etc. So again, you don't have to know, like as a microbiologist, we know a lot of the names of these things, but I'm not trying to put that on you at all. But we can kind of infer what kind of activities they might be doing. So this pipe may have failed due to a variety of different types of microorganisms. And as you can see, this is again very complex. We have lots of different types of microorganisms there. So it might not just be one type of organism that's actually contributing to any type of corrosion. So it's kind of a powerful tool, really. It's kind of like fancy microbiology where we used to, you may have seen where people would have colonies on agar plates, kind of get isolates. We don't have to isolate anything anymore. We can run this molecular method and get kind of a list of the organisms in our sample. From the DNA, we can also target other types of genes of interest. So we can do something called like functional or metabolic gene analysis. So when I was showing you earlier, this kind of diagram looking at c-Myc, for example, mechanism, we know that the gene that is really important in the process of converting sulfate to sulfide is encoded by this gene, okay, DSRAB. It encodes one of the main and really important enzymes in that sulfate reduction pathway. So if we really want to know, hey, how many sulfate reducers, or do I have sulfate reducers specifically in my sample, I can actually do the same kind of thing. I can target this gene, make millions and millions of copies of it, and then see whether or not that gene is actually present in the sample. So that would be like an example of a functional gene, okay? So we can do that type of analysis. So we can quantify it, which can give you numbers. So instead of using those bug bottles where you're really limited to culturing specific types of organisms, we can use more targeted genes that we know encode those specific enzymes that are important to these groups of organisms. Yeah, so that's just kind of another method that's commonly used, quite commonly used if you want to be very specific about certain types of activities. And I kind of wanted to show you an example. Actually, this is from a paper that, that Torben was a lead author on. And they were kind of looking at comparing, you know, they had sort of a, I don't know the whole story, I can't remember the whole story, but they were looking at, I guess, the top of a offshore oil production platform, some various locations that were suspected of having some microbial corrosion problems. And they actually did some counting as well, like using bug bottles as well as using qPCR to enumerate different types of microorganisms. So if we take a look at, you know, they had primers for total bacteria, sulfate-reducing bacteria, sulfate-reducing archaea, and methanogens. So you can see that there's numbers ranging from, let's say, 10 to the 2, in some cases 10 to the 11, 10 to the 5. So quite a lot of these different types of microorganisms. But when they use the bug bottles, if they had only used the bug bottles, you can see they had very few microbes in their system, right? 10 to the 0, 10 to the 1. So if they exclusively used the bug bottles, it wouldn't tell the whole story about the types of microbes that were there. So this was a kind of a case for, you know, we should be incorporating more of these MMM methods into some of the microbial analysis. It gives more information, probably more accurate information, because you don't have that cultivation bias. And so the third molecular technique that people use sometime, I think it's still largely academic in many ways. I know of one oil company that does, instead of doing that 16S sequencing to kind of get the list of microbes, they just do a metagenome on samples every year. Just is cheaper, they can do it, they have the people that can interpret the data, but it's kind of giving you probably more information than you want. So this is where you basically take a sample, you extract all the DNA, and you sequence everything. So you get a lot of data out. And it's kind of like one of my colleagues had a really good analogy. It's like if you went to the library with real books, you ripped all the pages out of those books, put them in a pile, and then you have to like put all of those books back together. That's kind of what you're doing here. So you're getting all the DNA, you're breaking it up, you're sequencing it, and you have to use all these programs to kind of put those genomes back together. So it's computationally quite heavy, but in the end, you can basically reconstruct the genomes or those DNA blueprints of all the microbes in your sample. So it doesn't just give you the names, but it also gives you all of their, you know, can they reduce sulfate? Can they degrade hydrocarbons? Do they have biocide tolerance? You can get all that kind of information. But it is very, you need an expert for sure to be running all of the bioinformatics, we call it, and then interpreting it in the end. But it can give you a lot of information. I would say it's still a little bit more academic. And this is just an example from a research group. They were looking at some Alaskan oil fields that had potential souring issues. And so using kind of a primer to target specific sulfate reducers, they ended up not getting that much information. But when they did a metagenome sequence, they were able to get a whole list of sulfate reducers that were present in a lot of the samples, as well as a lot of the gene information associated with those sulfate reducers. So it just gives you more information upon which you can maybe make some more decisions. But it is quite a lot of data overload. Okay, and so the last test that we're going to talk about is one that you will be working on today. And so this is kind of an enzyme-based test. It's called the ATP test. So as you maybe recall, ATP is kind of the energy currency of cells. It's present in all living cells, but it rapidly decomposes in dead cells. So when you do this type of an assay, you're basically estimating the number of active cells in your system. Okay, so it's kind of used as an indirect enumeration of living cells. It doesn't tell you what kinds of cells are there, but just kind of a number of active cells. It's definitely a technique that has gained popularity in many sectors in at least the last 15 years for rapid on-site monitoring of microorganisms. And so the assay, you kind of have a sample, you actually add an enzyme called luciferase, which produces light. And the amount of luciferase is correlated with the amount of ATP in the sample. You measure in a luminometer, and then the light output is basically related to the number of cells. And so people have done a lot of background work, like how much ATP is present per cell in an active cell, and you're assuming you have 1 femtogram of ATP per cell. So it's a very rapid field assay, and you'll get to actually do that today to kind of estimate how many active microbes are in different environmental samples. The nice thing about this assay, it actually comes in a suitcase, which I have here. So it's very field ready, and it takes about 10 to 15 minutes to run a sample. So it's designed to be done on the back of a truck, right? So if you're an oil field person and you're wanting to go figure out, your bacterial count across, many different producing wells in your field, you can just kind of drive around and do this assay on the back of your truck. So it's a very popular field method. So this little thing here, this is the luminometer. So you're going to be preparing a sample and reading it in one of these. Okay, and then we're going to enter your values into a spreadsheet, and it will tell you how many cells you have in the sample. So those are kind of the common ways. Just going to go back here a sec. Those are sort of the common ways that we can use to measure microorganisms when we get different samples. And that applies to not only interrogating samples for potential for microbial corrosion, but really any type of field sample. So if you're working in some other sector, you can use these techniques as well. Okay, not just oil and gas. Okay, so I'm going to talk very briefly about the importance of proper sampling. So remember my very first slide, garbage in, garbage out. So it's extremely important that when you're doing any type of sampling for microbiological analysis, especially for DNA analysis, that you use best practices. So for example, using sterile vessels and sampling tools, I have received samples in former like Coke bottles, things that people have drunk out of. They touch their human bacteria, into their oil field samples. So making sure everything is very sterile and clean. This is really, really important. Making sure you take the appropriate samples. So as you mentioned, biofilm solids are best, okay? Because this is where the action is if you're looking for corrosion. Sometimes water is the only samples you have. It could be because people haven't implemented, haven't put coupons in their lines, or there's just no way to sample any type of solids. So you kind of take what you can oftentimes. And if you're doing any type of analysis, it's always good to have, comparing, let's say, the microbiology in a non-corroded area versus a corroded area. So a little bit about having the most appropriate samples. And then also having appropriate preservation and timely shipping. So Torben just talked about a case where, you have a pipe, you're sending it for metallurgical analysis. A month later, it's been sitting out on someone's desk for a month. Now you want to do microbial analysis, and the microbiology has completely changed. So we've actually done a lot of studies to figure out what types of preservatives that you can add to kind of, freeze or sample or freeze the sort of microbial community at the time that you take a sample. And then you can ship it and keep it cold and analyze it a little bit later. And I do have one case study that we did just to kind of show how samples can change. Microbial communities can change over time in samples that are not properly preserved. And so I have microbial names on here, but you don't have to know any of them. But this was kind of we were sampling some offshore separator water, so on top of an offshore platform. And the water in the separator was at 55 degrees Celsius. So what that means is that the microbes that live there would be thermophiles. Okay, so they're heat loving. And so that would be organisms like Thermoanaerobacter there in the green. So when we took some samples and we properly preserved them for DNA extraction and kept them cold, when we sequenced the samples, these are just kind of a relative abundance of microbial names, you can see that most of the population was those thermophilic Thermoanaerobacter samples. But when the samples were not properly preserved, they just were kept cold, they didn't have a special preservative in them, you can see that just by looking at the colors, each bar represents a different microbe. You can see that had changed quite a bit. And so these were samples that came from offshore Newfoundland. They took 8 days to get to Calgary. And that's how much the microbial community changed, right? Just from not properly preserving them. So it's really, really important to kind of have a preservative in your sample vessel, collecting properly with clean tools, and then you can really trust the data afterwards, or the data at least makes sense with the environment from which it was collected. Okay. So as I mentioned, we have done a lot of work to kind of figure out, what types of preservative chemicals can be important and long-lasting for preserving samples for molecular work. And so we've now been able to integrate this type of information about the best practices for molecular methods and sampling into industry standards. So this is one that I mentioned at the beginning. It's a standard now that if you want to go collect samples from molecular biology work, these are kind of the best practices in that standard that you can follow. So a lot of our research that we're trying to do is ending up in standards that people can then follow. And we highly recommend that if you are ever in a position where you have to collect samples for this purpose, these standards exist. So you can follow them and you should be able to trust your data in the end. Okay. So I'm going to have a few slides about talking a little bit about measuring and diagnosing MIC in the field samples, right? And so we talked a little bit about, there was some discussion at the end of my last presentation, like, kind of diagnosing the root cause of failures due to corrosion, what kind of mechanism happened, especially microbial corrosion, can be very challenging. And that's because there's no single measure that can identify like MIC. This absolutely MIC happened, right? So we need microbiological evidence, chemical evidence, metallurgical evidence, and you kind of have to put the evidence together in order to maybe, come to the conclusion that microbes may have had, contributed to a specific failure. So multiple lines of evidence is pretty important. And ideally, if you're able to get an uncorroded sample or a sample from an uncorroded area versus a corroded area, that is definitely ideal. And again, one of the reasons is that, you have microbes in a system, they produce products, those products affect the materials. In turn, the materials, might have some products associated with them. The media could have specific compositions that will then determine the microbial community. So there's all these interactions between the metal, the chemistry, and the microorganisms. So nothing's ever really straightforward. So for chemical measurements, you'd want to look for things like volatile fatty acids, like acetate, propionate, butyrate. These are sort of known metabolites produced by acid producing bacteria that seem to play a role in some microbial corrosion. Salinity matters, pH matters. You have all your different potential electron acceptors. We've assumed here anaerobic conditions. And then you want to look at the elemental composition of any corrosion products that might be on the surface. And generally, these are mostly iron and sulfur related measurements. So you might be looking for iron sulfide, iron oxides, iron carbonates, etc. So a lot of these can kind of give you some clues as to what types of microbial corrosion may have happened. And then you can also take a look at the metals. So I know like you have done a lot of, there's many different tools that can, kind of do an analysis of the metal itself. So you can maybe look at pitting, which is often, but not necessarily distinctive of MIC. Pits, any pits that you see should correlate with other measurements. And you can use a lot of different visualization tools in order to kind of see what effects the microorganisms might have had. And so these are just, again, some bead pictures. We use these little tiny carbon steel beads that are nice and shiny in sterile controls. We did an experiment where we added an inhibitor and a biocide. So at the end of the experiment, also nice and shiny, but when there was no treatment, the beads were all kind of chewed up by the microorganisms. So very, very rudimentary microscopy here, but just a reminder to also take a look at any appearances of the material itself. So combining the microbiological tools, the chemical, the chemistry of the environment, and the metal, the effects on the metal. Really, really important. In the microbiology world, we like to set up microbial corrosion experiments. So let's say, we have a case where we've done these various, chemistry, microbiological, metallurgical analysis, but we managed to get a little bit of material, and we want to kind of do some experiments in the lab to see if we can identify what type of microbial corrosion happened. So we can set up laboratory incubations with potentially corrosive field samples. So that could be water, pig solids, solids collected from a leak site, or those corrosion coupons. And then we can set up different types of incubations under static or flowing conditions. So a lot of times in my lab, we do anaerobic work. So we set some sort of anaerobic bottles that have a corrosion coupon in them, let's say, or some little carbon steel beads, put them in holders. You're going to see a system where we have some dangly coupons. But anyways, we can kind of set up some of these bottles, either statically, or we can kind of have like a Robbins device type flow system where we can measure, have flowing, medium kind of flowing through some corrosion coupons and then monitor them over time. And so with that kind of information, we would use different types of corrosion coupons that can be coming from any type of metal. You can kind of buy these from different vendors. And we also have used these little carbon steel ball bearings. Well, they're not that big. They're actually really small. And that's so that we can get we can use these kind of little metal beads as a kind of screening a whole bunch of different samples for corrosion. The nice thing is they're uniformly sized. The negative is we can't really do a lot of good surface analysis on them. So in our experiments for corrosion, we often just use the weight loss method because that's very, very simple. So we would weigh our pieces of metal before the experiment, let some days pass, process the coupons and weigh them after, and then we would enter this into a corrosion rate calculation, and we can determine a corrosion rate, a general corrosion rate. And so there's industry standard protocols for preparing metals before and after corrosion experiments. And so you're going to, in the lab this afternoon, are going to take some, kind of do some of this procedure to process those pieces of metal. And then you can certainly do electrochemical measurements. I'm going to leave this alone because I know very little about it, but you can definitely do some electrochemistry measurements. Yolanda's lab is doing that quite a bit. And I will embarrass myself if I try to talk about it. So, but anyways, that's definitely another way that you can measure corrosion. It might be a little more challenging with complex samples, but I think you can still probably do that. If you want to know if a certain mixed culture or mixed sample is also corrosive, you can use these electrochemical measurements. In my lab, we're not very sophisticated, so we kind of just look at the weight loss of the metal. Okay, so this is just an example of what a coupon that kind of came out of a sulfate reducing culture. You can see it's got some black and very, very slimy. So we can kind of get rid of that layer to kind of look at the underlying metallurgy. And I know it's pretty hard to see, but there's actually some evidence of pitting on that coupon over there. Okay. So what we would do, as I mentioned, we would take some sort of culture or sample from the environment, add some pre-weighed, these are beads in this case, incubate over time. In this case, we see some black iron sulfide formed, clean and remove those coupons and weigh them, and then kind of look to see what those kind of look like under very simple optical microscope. And then we would weigh them, and then we would calculate a general corrosion rate to see, have the microorganisms reduce the weight of that metal. And again, it's always compared to a sterile control where the microbes are not present. This is kind of a formula that just comes from an industry standard. And so we do have this formula in the lab package, and you are going to weigh some stuff. Yes?
Last picture there. So I see your beads, they're kind of in the corner of the bottle. Have you done any controls where maybe you have them on some sort of pedestal or some suspension in the middle of the solution?
So not with the beads we haven't, but we do that with the coupons. So the coupons are not lying flat on the bottom. The beads, not so much. Yeah. So yeah, there's always a little bit touching, right? Yeah. Yeah. Yeah, we mostly use the beads to kind of scan, I guess, or survey different types of samples for corrosivity. And then if they're corrosive, we will do better experiments. Yeah, we had a project where we got so many samples in that we needed to something kind of quick. And that's why we ended up using these little beads. Yeah, so just in summary, several methods can be used to determine the microorganisms part of MIC. And again, this is just one part of the MIC story. Really important to obtain high quality samples. And so again, if you didn't know already, there are many nice industry standards that are available to carry out those best practices for collecting and processing samples. And then MIC experiments can be further set up in the lab to determine the corrosivity of that field sample. For us, again, we're looking for microbial corrosivity. So you can measure general corrosion rates, and super exciting, you get to kind of try this today if you haven't done that work before. So yeah, so that's all I have to share on the SOPs and the.
Would you help me get the slides up?
Yep, any questions?
Yeah, any questions?
Yeah.
Also, do you also pickle away the oxide or just clean off the biofilm?
The oxide should go away as well, because the cleaning procedure gets, uses acid, acetone, like a variety of different washes to get rid of the oxides. And so we do try to, before we do the microscopy, we try to put them in an anaerobic atmosphere as well. So we don't have a lot of like oxygen, like rust happening onto the beads.
Proxy.
Yeah.
Because normally when we do weight loss, it's really important to choose the right pickling method for the right type of.
Okay. Okay.
But here's probably a good proxy.
Yeah, we've had the problem before where we've, sometimes our coupons or beads weigh more than our starting weight, right? So we haven't been able to get all the residue off of the,
Exactly. So
yeah.
Difficult to dissolve oxides, right? That can.
Yeah. Yeah.
Either from the material itself or in that.
Yeah.
So
yeah.
You have to use a pickling method.
Yeah.
Increase temperature or whatever.
Right. And I would say most of our work so far is just HCl and yeah, just room temperature stuff, which is the procedure you'll use today. But yeah.
This area will also kind of help with any gunk that still remains on.
Yeah.
Also like doing multiple samples and getting statistics on it.
Absolutely. Yeah. Like we would set these up in triplicate at least. And then we weigh all the little, the beads themselves are replicates of one another. So we just weigh them all together though. So. But yeah, I would love to know about other pickling agents that we could try. Yeah. Okay. Okay.
Where you have like all the materials and you get, but it's probably only one option. Oh no, there are several options for like mild steel or normal.
Okay. Yeah.
Try different, but you also always have to observe. So it's, yeah. We did recently a study where, I mean, this is a typical question from the reviewers, right? Is the pickling working? Is it
okay?
Or is it under pickling?
Okay.
And, so we did a study where we compared a number of different analytical methods. Like we tested everything in solution. We did mass balance. We had different techniques, and we were then measuring all the different mass loss for 3 different techniques, and then estimated if we underpickled or overpickled.
Okay.
So, but it's always a question. This is why. But
yeah.
It's such a big difference, right? So it would.
We often see like a lot of like the beads are just chewed up. Yeah. Yeah. And there's also cases we've had where the medium itself is more corrosive than when the bacteria are there.
Yes.
Especially under high saline conditions. We've been doing some work under 2.5 molar NaCl. And yeah, with the salt itself is corrosive, and the bacteria seem to actually protect the metal.
I've also had those cases.
Yeah. So it's very interesting to see those observations. But yeah, I'll take a look at the paper with the comparisons and the standard. Yeah, that would be great. Thank you.
More questions? Yeah, I sent an email, so we'll take it. All right. While Lisa is getting the slides up, the last bit between you guys and lunch, that's failure analysis. Has anybody been involved in any failure analysis cases? Yeah. What? Just tell about it, just quickly.
I'm
Just speak up.
with the coated zirconium alloys, it's failure analysis based, especially if you do like full bundle assemblies of it, where you run in high temperature steam and steam where your coating has failed and why it's failed at that particular point. Go, you understand what your processing conditions are, understand what the base material is like, and then you go through the whole process and see it's failing here at this site because there's a coating defect or something.
Okay. And you?
I did a publication with the Canadian Nuclear Laboratories. They did a test loop of molten salt.
Yeah.
That was intended to run for a year, lasted a week.
Wow.
The whole step of the failure analysis was I got sections of the tubing from different current. I moved the salt. In the cold leg, they just had a massive deposition of a lot of corrosion product containing a lot of iron and other spinel-like structure. But in the hot leg, it also showed a mass amount of dealloying and dissolution up to about it was a few hundreds of microns. So if it didn't fog up and blew up there, it would have probably broke through the wall in the hot leg.
Yeah.
But the process was that a big thing is that always try to do everything you can before you start cutting and sectioning because you can't go back.
Yep.
So a lot of the start of it was just taking regular images, like putting stuff under the microscope. That's like good. And then going on to the more serious like SEM. Part of the paper also went into XRD and TEM. I didn't do those parts, but I focused more on PSC.
Do you want to come up here and do the lecture? You no, but that's great examples. Any other? Yeah.
Different, like one was a heat exchanger that failed in a company, another was a stainless steel truck that was not cleaned properly and they didn't know why that was pitting. And then there was, I did a lot of analysis on implants, so failed implants, 240 and others. So there we had a little bit of MIG as well in one case. And there is one of my mentors, Paul Linhardt in Austria. He does a lot on MIG failure analysis. So he goes to the plants, and that is the
hydroelectric
plants.
Yeah.
And there he put an electric sensor on one of the corroding turbines. And the funny thing was that the potential was changing over day and night time. And he figured out that those algae that were actually, so there are really cool things you can do in the big area because it's every case is new, right? Yep. And failure analysis. So one common thing for all this different failure analysis is always hard to get the real information from the people that are responsible because they usually hide. There's usually a question of fault, right? And then once you actually figure out, could it have been something? Then they come with it. So it's like an iterative process.
Yes. I was just to say that there's the whole technical aspects of doing failure analysis, like the methods, the protocols, the procedures, but you really need to give yourself the Sherlock Holmes hat on and be a psychologist and talk to. Exactly, it is like CSI, and I think that's very intriguing. You don't know what the root cause is before you start out. You kind of keep it open, like as an investigation, and then you start questioning. And actually, you get more than half of the answer just having a cup of coffee with these people, and then start putting questions out there and get them to talk and take notes or record it. So these are things that you cannot plan. It always happens Friday afternoon at 5 o'clock. That's when they call, because these are operations. They run and they produce. But I'm going through a case very detailed. But I think this whole thing about being CSI or Sherlock Holmes, that's kind of your job as an investigator. So let's see how we go. First, maybe just sit 2 and 2 and think about what you need to conduct a MIG failure analysis and get a few things down on paper. And people online, they are also welcome to put up stuff. So just maybe tune in and do a little bit of thinking from what you've learned this morning. So you can sit 3 and 3 if that makes sense, or maybe one can go down to the gentleman in the back. That's a lonely girl as well. So you team up. So spend 5 minutes on that. What's the exit? What's the exit? I love that. That was hilarious. Maybe jump to these people here.
Oh, I think that's.
To
what are the procedures concern preservation, maybe
yeah, so like knowing
It's just like the basic water compensation, science, what's in it.
then they would also stop all quotations that and growing. So if that was up switch off the temperature,
because
why they have to failure and switch off the temperature, then you cannot, you have to get the samples as soon as possible, because otherwise it will change. So then that is something special for make.
So yeah, samples right away, well preserved.
Yeah, and you need to know the preservation.
Yeah, it's like very
Yeah, yeah.
Before.
Yes.
data or something like that.
Well, I think what often happens, like I know with pipelines, they sort of dig it, and then they don't have any microbiologists around. Like turbines. That could have been very useful, but it's been very challenging to get the word. Oh, if we see this debris that might be slimy. The microbes could have.
Substance or whatever is just
I think we have a couple of champion that operate, pipeline companies like Enbridge that keep their own, which is nice, but not everybody does it. So yeah, like, but rapid sampling
to basic.
is possible.
Because then other like in
If you don't.
here it doesn't have to be, you need samples quickly. So that is repeatable.
I think many might do.
وقتی failure suppose removed root cause.
Yes, many different.
Yeah.
Yeah, like you said, getting the operational data.
چی باعث شده که تو این
Golden.
فیلیر رو داشته
A lot of people are.
در همین سوال دانش بیشتر من نمیشه فقط با اینکه تو آب داری و مثلا محیط چیز غنیه بگی که 100% این میتونه به هر مدلی که
Some amount of knowledge or like
Like,
yes no
right, and that's where the.
structure itself,
داشته یا نه
microbiology.
اگر میک داشته من دی ان ای میگیرم
Okay. Should we try and get the discussion up here? Any thoughts from you guys?
Well, what we put as like kind of data would be useful is to know if what is the flow in the water, is it stagnant, what is the pH, salinity, what is in the water, whatever. And after there was kind of the basic of what is in the water bacteria-wise, is there something bacterial, how much is it? And yeah.
That's good. Well, good points. Yeah, you guys?
We got the second question to answer. We think firstly the data should be repeatable. Then we can, according to the standard procedure, we can get different data from the experiment. So it's important then repeat another experiment.
Yep. Thank you. This group here.
the main thing, obviously, that was hashed a lot, and is very important, is that you need to have a good sample to start off with. And also having that MLOE of having multiple people to come together, not.
Yeah.
And then in terms of like what kind of knowledge and data you need, again, it falls back onto that like multiple streams of thinking of like having just that basic surface, but also people that can sit down and think about an actual mechanism and also getting like you.
Yep. Thank you. You guys?
What kind of mechanic?
Environment, how the environment is affecting it. Do we have any like oxygen there or stuff that is broke?
Okay, cool. You guys?
We're thinking a lot about how quickly you need to get samples.
Yeah, yeah.
That's, especially if the operation is shut down and maybe the temperature is changing and so on, then also the microbes will change.
Yep. So timing is important. So a lot about sample methodologies, reproducibility, getting the right team set, different lines of thinking, DNA, different types of analysis again, and the timing.
It's getting a lot of questions.
Yes. These are all really, really good points, and I think we'll get through those. So I'll start out, why do we do like failure analyses, and then go through some main steps. And failure analysis can be really, really broad. It can be a chemical plant that explodes, or it can be a plane that crashes, or it can be the sewer of Calgary, sorry, the water, drinking water of Calgary, main lines that are collapsing. It can really be a lot of different things. It can just be your regular blender or kitchen machine that doesn't work. But I think you're getting the point. And then I'll go over a case study from the North Sea in Denmark. So a little bit about motivation. We want to get to the depth of what the true corrosion mechanism was that caused the damage, the contributing causes that led to the corrosion mechanisms, and also the root cause that failed to identify or control the corrosion damage. So we get out to the failure site, we get out, there's something that happened, or a pipeline is leaking, we have oil all over. So bring a team together, take good pictures, take a step back, get an overview. You've probably heard about these laser scanners that can scan the whole crime scene, and then you get a representation for later work, because the runway needs to be cleared, like airlines need to go, and we cannot stick with this for weeks. So you need to make a representation of the crime scene. Identify regions of interest, need for preservation. That was about the timing that Yolanda mentioned. Yeah, some measurements and 3D scanning. There will be some documentation about the materials, drawings, some incident logs, procedures for repair, maintenance history. Has there been anything similar to this? Maybe Water Systems of Calgary saw this 20 years ago. I don't know, I'm just speculating. Are there any sensor data? Are there any surveillance videos that you could look at? So has there been anybody coming to the site and doing some like terrorism? Is it not really the stuff, or sort of the equipment itself? It might have been somebody from outside. Weather data, like was it raining? Was it like high winds? So there's a lot of things that you can look at, and this is basically also what insurance companies they are looking at. When you claim that I got late to my flight because of traffic or something, they look up, yeah, there was actually a lot of traffic that day. So then there are the non-destructive tests. So you talked a little bit about that. You need to do a lot of stuff before you start destroying your sample. And again, photo documentation is really important. Different types of methods that can be used. Ultrasonic, you can scan the outside of the pipe to look at wall thickness. It's really intriguing what can be done non-destructively. Examination of the surface itself. There's some cracks happened at a well site. but you can use scanning electron microscopy, EDS, XRD, particle analysis, different kind of scanning. I added in microbiological data. That's going to be part of the investigation I'm going to talk about in a minute. Testing of material properties. So now we're getting to the destructive part. Chemical analysis, mechanical testing, tensile testing, hardness testing, corrosion testing, and so on. So this is where you go more into the metallurgical properties of the material. These become very important if you have welding, heat-affected zones, base material, and all these kind of things. Metallographic examination. There's a standard listed here. You can even use like regular light optical microscopy. Yeah, so here are some nice photos from the metallographic examination that is used to determine the soundness of the base material versus the welds. Check heat treatment and cleanness. And I'm not a metallurgist, but I consult my good friend Rick Eckert when I have stuff like this, and he's a metallurgist, so he can tell me what's up and what's down on these photos, and I can tell him about the microbes. So a lot of stuff can actually also be like taken out in the field. If you can't bring the pipeline into the lab, you can actually take a lot of this and bring it out into the field. So there is like the existing assets. So there's a reasoning, and also there is future assets. We really want to learn so that we get to a better point. We want our assets to be sustaining longer, and we want to build them in a way so that they have a longer lifetime. But actually, the existing assets, you could use this knowledge to select and optimize better mitigation measures, plan inspection and repair, and use the most appropriate testing and monitoring methods. So there's really a lot of learnings in this, and when you really get into the weeds of it, I think it's super interesting. And then we get to this figure again, and you have the paper that we published. We actually had a graphic designer doing all the figures with the puzzles getting together. And everything that I talked about previously is really well described in many companies that does failure analysis. But they kind of been missing out on this part here for decades. And I think there's a reason for that. How many of you guys have had a microbiology course? Yeah, I know that you had. And there's a biologist in the back, and Mayet. So there's 3 people, and you're chemists, and you guys are microbiologists. But engineering, nah, we do our own stuff, but you need to know about this. And that's been a blind spot on the map for decades, for many reasons. And I can talk for hours on that because I work with engineers, so I know some of the stuff where the barriers are. But if you don't know about it yourself, you just need to call a colleague that knows about it. And there's Lisa, and there's me, and there's a lot of other people out there that will be able to help you. And it's a service. Like, it's a service that you can purchase. And also, I think part of this is that we've been lacking good standards. So when something is in a standard, people can look at it, okay, it's in a standard, then we have to do it. Because if something happens and the legal system comes and tells you, you have not been running this according to best practice, we either fine you, or we put you in jail, or like something nasty happens, or we close down your business because you've not been running this according to best practice. And that's something engineers understand when you close down a business or you put people in jail for not obeying like latest practice. So I think it has a lot of reasons, but now we have these methodologies into standards. So there's nobody that you come and say, I didn't know. Yeah, then you're not up to speed, man. So we try to put a process together where we actually integrate the microbiology. And you will see this multiple line of evidence again. So assemble, organize what we have, and then comes the integration part. That's the hard part. That's where we need to put it together. Then we're going to look at spatial differences, corroded versus non-corroded. So we have two different areas where we have corrosion, where we don't have corrosion in the system. We look at design and operational contributors, consider possible mechanisms, conclusion and supporting facts, data gaps, like what additional information would you like to have? Sometimes they just come up with two numbers and they're like, so what's the root cause? It's like telling what the weather is going to be in 10 days and you don't know what the pressure is, what the wind direction is, what the temperature is. You have no data point. Then I can't tell you about how the weather's going to be. It's simple as that. So many times you have to go back and tell the people, I need more data. I need something to work with. And then these are the important parts, the recommendations for onwards operation. So we tried, it's actually the book, chapter 3 that you got. You can pass this one around. Just make sure it ends up with Lisa Gieg because it's a present for her. You can flip the pages. It came out in April this year, and I put down the logo in the corner in Euromec. So it's sponsored through that program that we had running for 4 years. And in chapter 3 in the book, we actually go through this process. And it's widely described in an earlier book, like with all the details and so on. And then we took it and then we made the sort of the process around it in this new book. But yeah, this book is failure analysis of the MIC. So we're looking at a leak in an offshore water injection system. And this is the platform, which is called SERI production platform. It doesn't have any pipelines connected to it. It has a storage tank at the bottom of the ocean where the oil goes, and then there's a tanker coming once in a while to pick up the oil. So that's it. So this offloaded to an oil tanker. This is the main production platform. There's 2 satellite platforms. You can't see that on the picture here. So 32 kilometers away on each side, you have satellite platforms. They are non-manned, but you actually have water going there so that it can be sourced into the reservoir to keep the pressure up. And then they came up with this brilliant idea. Let's start. I have somebody that's texting me on my watch, so I'm just going to put my watch here. They had this brilliant idea of mixing seawater and produced water. And then they had a leak of the pipe, but that's actually 10 years earlier than this case. So they kept on dragging these problems along. But they had a leak in 2017 in the top side of this production facility. So I think Monica, she talked a little bit about corrosion circuits yesterday. So that's a way to break up the different processes and compartments on the platform. And there can be like, say, 200 corrosion circuits on the top side of a platform. So this is a schematic of the seawater. It's pumped up to the platform with some booster pumps. Then the water comes into a deaeration tower. It's where the oxygen is stripped off by a chemical process. And then that or deaerated water goes to the water injection booster pumps. They add some biocide, and then it's injected into the reservoir. Very, very simple schematic. At this point, they had a leak right where you see the arrow. So we cut out 2 pieces, X1, and that's where we had the leak just at the bend. And then we cut out X2. That was an area with no leak. So we had a corroded side and a non-corroded side. And that was sent to shore. This was what it looked like. So you see this is X1 with the bend, and they put a clamp on just to keep the pressure in, because it's like a high pressure system, and they had like water sprinkling out. And here you have the X2, the non-corroded one, with the flange at the end. So these were the specimens. Problem was that they've been taken down. It was already dry because they shut off the production, so it's already dry. And then they put it on the deck offshore and mounted it. They put it on the ship, and the ship had to go to the shore. So within 3 weeks, I came to the shore base, and that was where I got access to the samples. So they were already like 3-4 weeks old and pretty dried out at this point. But this is what we have to work with. This particular carbon steel grade that you see there has been in operation for 18 years. You see the diameter. It has been produced water and seawater mixed over those. Yeah, you go.
Like, yeah, when you show up to this, do you have like a timeline, like engineering change forms where they did some change to this section of pipe or whatever, or how they operate this pipe? Do you actually have a timeline of?
No, that was one of the things I would like to have had, but it just did not exist. It might exist in an Excel sheet in an employee that left the company 5 years ago. I have no idea. There was no way they could dig out that information. This is the water, how it was treated. And you will see in a minute when we take off the wrapping here, the clamp, the coat comes out. You can see the coat has come out a little bit, but the coat comes off, and then you can see the hole from the inside and from the outside. But that's some of the information that was just missing. And then we just need to characterize that as missing data, basically. So here we're looking at the base. The flow is this way. We have the leak up here. There are some other corrosion cavities, and there is a weld root and misalignment. This is where the weld is. You'll see it in a minute more. This is looking from the outside. Here you have the pinhole, and you can see the paint has come off. This is a little bit closer. So you see like 5 millimeters in diameter. This is looking from the inside and out, and you can see it's pretty cavitated right around. So we did a lot of metallurgical analyses. And again, I'm not an expert in this, but I was told that there was really no problem in the type of the base metal as compared to the welding heat affected zone. So we couldn't put a course on that. EDS, so energy dispersive X-ray spectroscopy. We did it at a number of places. So actually we had the brownish layer, which was kind of like the part that is oxidized. So, and then we scraped into it, and that's the blackish layer. So just keep that in mind. There's 2 types of analysis here. Sample 1 and 2 would take it on the blackish, and 3 and 4 would take it on the brownish. And these are the analyses. 1 and 2 are the black, 3 and 4 are the brownish. So we don't have any XRD data to determine the mineral composition. So that's another area where they could have increased the value of the analyses. So here are some of the components or elements. The iron are likely oxides, possibly sulfides. You have silica, likely comes from sand. But yeah, this is what is closest to the metal, and this is what's closest to the liquid. And you see an increase, at least on the elemental sulfur. So now we come to the samples for microbiology. So we had this is really close to the leak point. So I call that at the leak. And we had 1 sample upstream and 1 sample downstream. But you can already see how dry it is. So it's really not an optimal sample for this. But we give it a try. The good thing with PCR is that in theory, you can actually, as long as you have DNA there, you can actually amplify it. You probably heard about mammoth, they dig out of the ice in the tundra, and the Neanderthal man, you take bones out and you can actually do DNA analysis on ancient DNA. So it's doable, but it's not the best type of results that you will get. So yeah, it's a little bit about the timeline, but bottom line is that the samples were pretty degraded when we got it. They didn't preserve the pipes well. It would have been nice if they had sealed it off with plastic to keep the moist in there. They didn't do that. It was exposed to the air and to light. So if you remember the three areas at the point upstream and downstream, we targeted different types of bacteria with PCR. And the overall message is that at the leak site, we had the highest number of total bacteria. And we also were able to discover or measure archaea and some methanogenic archaea. All the others were pretty much not detected or below detection limit. But it tells us that there was more activity microbiological-wise at the area. And we find some of these usual suspects that are causing the corrosion normally in oil and gas. Good thing is that they have done monitoring programs on water over the years, and we got access to that water data. And there we actually find sulfate-reducing bacteria and methanogens. So that was done on fresh samples. So now we have all this data, and now we need to put it into the framework or the process here, and then start asking questions and try to connect the dots. So this is a bit overwhelming, but it's actually taking those 4 compartments in the previous one, chemical, materials, microbiology, operation, and putting it all on one slide. So we know that they started to mix seawater and formation water. There's a low concentration of H2S and CO2. They had corrosive compounds in the water. We didn't observe any oxygen. pH was around 7. And the produced water that has been running in this system can carry organic carbon, which is an energy source. We found localized corrosion in pits. We found scale formation, sand, other deposits, composition of inner and outer layers are different. So the black and the brownish. Presence of sulfur in corrosion products. Minerals form is unknown. Could be sulfide, sulfate, or elemental sulfur. And then we have the microbiology. I added that water samples had SRBs and methanogens and stuff under operational. They mixed, but there was no record. They had actually dead-end piping in the system, so back to the stagnant. And the temperature were favorable for microorganisms, like 10 to 50 degrees. So that's all the evidence. Yeah, you go.
It was stagnant at periods, but I'm sure there's periods of time where it's actually flowing. What in that portion? Because you're between 2 pumps, so I'm sure there's.
Yeah, but it was pretty high flow. I can't remember exactly the velocity.
The flow-induced corrosion.
No. I'm coming back to that in a little bit on the bent. Yeah. So these are some of the questions we ask and try to come up with evidence. What did I do?
Nice. Cool. No action is required. This is typical Canada.
No action is required. Take a coffee.
Was it expecting the zones?
I think you just acknowledge it on the panel.
I have the mark.
Acknowledge and close. I'll tell you this, on your phone, you can get these alerts. And I was in BC traveling with my wife in the mountains. And I've never had a flash alert like that. And you know what it said? So we were out in the mountains in BC on a holiday, just came from a winery, going to Vancouver. There is a serial killer in the area, just to notify you. And then we came to Vancouver, some like treetop stuff, and we kind of walked around, who's the serial killer? And we asked somebody that we thought was not the serial killer, what does this mean? Oh, it happens all the time. Okay, great. Yeah. Anyway, back to this. So these are the questions we ask and try to come up with some supporting evidence. So are there chemical indicators that could suggest or support microbial activity, blah, blah, blah. And yes, we saw methanogens in our assays, and we have CO2 there, and we have organic material. So they can actually live there. And again, we saw SRB that was in the water, and sulfate and organic matter is what they live from was there. So that's plausible. Are there some of these conditions that are supportive for microorganisms? Very supportive. Are nutrients present indeed from the water and the material? And we just go on. Which chemicals, so now it's the chemistry and the corrosion that we're trying to integrate. Which chemical species are associated with the corrosion products? Some form of sulfur, could be from sulfate in the water or sulfide. We need their XRD to determine. To this one, no oxygen, temperature indeed significant, and which abiotic mechanisms could result in the observed corrosion. So on the next slide, we discuss various types of corrosion. I'll show you that. We use this scheme. So which one are possible in terms of mechanisms? And MIC has its own, because it's split out into more mechanisms. But yeah, H2S is possible, elemental sulfur on the deposit corrosion, and then metabolite MIC. So that was one of the mechanisms that Lisa was discussing early on. So you see there's a process as we move forward. Spatial differences. So the corrosion was local at the bend. Significant corrosion had occurred in a number of bends in the water injection system, while less corrosion has been observed in the pipe sections. And then we came up with this, that the curvature of the bend could affect the local flow conditions around the well route. So that was what you asked about. Again, there's more mechanisms to it. So it's just not just the one thing. The highest abundance of a bacteria was in sample A, which has taken close to the leak point. Operating, the blending of the produced water and the seawater, increase in temperature over time. Bends affect the flow around the well. Metallurgical differences between the pipe and the bend and the wells were not considered to be a significant contributor. So this is, I think Lisa had a similar thing here. But this is chapter 4 in one of the books that I described. And these are the usual suspects, the methanogens and the sulfur-producing prokaryotes. That is dragging electrons off the metal surface. So for us, it was not a big surprise, but we needed to put all the evidence together. So gaps, we talked about additional chemical environment. It would be nice to have this at some point. Operating history of when the blending was, water chemistry data, sequencing of the microorganisms to determine if microorganisms, what kind of microorganisms it was. And I'm sure they could come up with a lot of extra things that they needed. Okay, I'll just put these up. What should the pipeline operator do with this knowledge? So they really should have a stronger monitoring program, also looking at CO2 and oxygen in the injection water. And established a program for biocide injection, and the separator and the coalescer and the degasser. They need to measure the microbiological activity, because that's where the bacteria flourish in this system. So taking it all down to a few takeaways, the corrosion failure analysis can provide valuable information to managers to reduce risk. And this multiple line of evidence should be enforced, and they should follow this structured process and consider both abiotic and biotic corrosion. Yeah.
Just like a general thought I had, because like piping, you have long periods and you don't know exactly where the mic's gonna put, but maybe you have a CFD model on that piping when you designed it.
Yep.
Give you information on where these dead legs might form and stuff.
Yep.
And then, like, could you then survey those areas with like a radiography or something like X-ray, and like see if this deposit is building?
Yep. They do that also with the thermometer, like thermographic identification, because the sediment will be colder. So there's all kind of means that they do that, and that's also what they feed into the risk-based inspection programs, where to look. But dead leg is always like a red flag. Let's go look there. Yep, good points. So I'm coming to an end. I just want to flag 3 things I like when having presentation to pay a little bit forward, and you can all get the slides if you like approach me somehow or through the coordinator. We did this book. It's more than 500 pages, and there is really a lot of information on failure analysis in all kinds of systems. So for us, it's become a little bit the Bible of failure analysis in water systems. There is sprinkler systems, there's drinking water, sewer systems, oil and gas. And it's just a great inspiration. And I think actually the online version is not that expensive. This one that I passed around, I hope it ended up down with you, Lisa or Will. It's open access, so you can just get all the chapters for free. As I said, this came out in April, so it's just fresh. And there's a lot on standards, on processes and ways to look at this. And it's really the experts of the MIG world that did this. And then if you want to take another course, we're running online courses with the University of Tulsa in US. So it's myself and Rick Eckert. And we've been setting up a course on failure analysis with them. So actually a lot of the big major companies, they send their people to these online courses to be updated. Yeah, questions? Yeah. Did any of you get to look at the video? So now you know what the bacteria they're sitting there with their drill hole. Yeah, it's a lot of information, but I think it gives a good overview, at least. Now it's the food, and then it's laboratory. So I'll put it over to you. Are there any questions? So was that kind of clear? Cool. And we stick to time. And if you have any questions, I gave some of you my card. You can just email me or LinkedIn me or whatever. Yeah, always there to help. And if I don't know the answer, I'll send it off to Lisa or Jason. So.
After lunch, we go together, right? From here. So we go at 1:30.
It's for 1:30. Yeah.
Yeah. Okay.
I think we're gonna go like at 1:15, our team, just to make sure we have everything ready and set up. Should be mostly ready and set up, but.
How do we find the room?
It's one up, isn't it? Or two?
You guys basically well, around the corner. I'll send you the
Okay, good. So yes, so for the online people, we will finish this.
Take this off.
Recording will be in that sense. We put all the recordings from all the days on our correct webpage, and we will put that on. Can we get the sides? Okay. Online.